Hello students, my warm greetings to you all. Myself, Miss Pranjali Mahendra Gaukar, Assistant Professor in Botany, Government College of Arts, Science and Commerce, Sakri, Goa. I welcome you all to the program in Bachelor of Science, first year, subject Botany, semester 1, in course BOC 101, course titled Biodiversity 1, which includes microbes, algae, fungi, and bryophytes. The title of the unit is Fungi and the module name is Reproduction and Classification of Fungi. This is the outline of my presentation. It states type of reproduction in fungi and classification of fungi. At the end of this module, you will be able to explain and elucidate upon the different modes of reproduction in fungi and explain in detail the Ainsworth classification of fungi. Introduction. Reproduction is a fundamental feature, and from this process, the new individuals and offsprings are produced from their parent or parents. The different modes of reproduction are vegetative reproduction, asexual reproduction, and sexual reproduction. In vegetative reproduction, it is the involvement of somatic portion of the fungal thallus where new individuals are formed without the production of spores by meiosis or by syngamy. So, this includes the following methods. Those are fragmentation, fission, budding, oidea, chlamydospores, rhizomorphs, and sclerotia. In fragmentation, it is the mycelial fragments are formed from the thallus and they grow into new individuals, where the fragments result from the accidental severing into the bits or fragments and through mechanical injuries. There are small fragments or segments of hyphae formed. Each segment, by further division of cells and apical growth, develops into new mycelium under favorable condition. For example, in mucor. Next, we'll see budding. Budding, here we can see in this diagram a small outgrowth called bud is formed, and from this bud the new individual cell of fungi is formed. Here the typical example is Saccharomyces, which is yeast. In fission, the cell breaks into two daughter cells and splits to form new individual cells or divided cells. Here the example is Zygosaccharomyces. In oidea, these are individual cells formed on the breaking of hyphae. For example, in Rhizopus. So this diagram shows the oidea formation. Next, it is chlamydospores. These are one-cell fragments which function as perinating bodies, where perinating bodies means under unfavorable condition they remain dormant, and after getting the favorable conditions, they germinate to form new hyphae or new fungi. Example is Ganoderma. Next is sclerotium. In sclerotium, it is a compact mass which is surrounded by a hard covering called as rind, and then it forms into the new individual. Rhizomorphs. They are rope-like structures in the interwoven somatic hyphae. An example is Amarelliella. Here, in this diagram, we can see the rope-like structure of hyphae. Next, we move on to the asexual mode of reproduction. Here, the spores are formed by the process of sporulation, and the spores are called as mitospores as they are formed because of the result of mitotic cell division. The mitospores produced in fungi are of three types, namely sporangiospores, zoospores, and conidiospores. Sporangiospores are produced inside a sac-like structure called sporangium. Here you can see the sporangium. The hyphae bearing a sporangium is called sporangiophore, and this is a sporangiophore. For example, in Rhizopus. Zoospores. These are motile spores lacking cell wall. After swarming period, that is the period of migration, it secretes its wall and germinates to form germ tube. For example, in Phytophthora. Next is conidiospores. They are non-motile spores and are single separate cells. You can see here the conidia. They are born on conidiophores and they may be simple, branched, septate or aseptate. Example is albugo. Next is sexual reproduction, which involves male and female gamete. The sex organ in fungi are called gametangia, and this process involves plasmogamy, that is the fusion of protoplast, karyogamy, fusion of nuclei, and meiotic cell division. The planogametic copulation is a type of sexual reproduction. It takes place by isogamous, oogamous, and anisogamous types. That is, in isogamy, a similar kind of gametes fuse. In anisogamy, two dissimilar kind of gametes fuse. And in oogamy, one motile and another non-motile gamete fuses. Here, the example is Olpidium. The next is gametangial contact. That is, through a contact or through a tube, the nuclei inside the antheridium, the male gamete, migrates into oogonium. Here is the diagrammatic representation and the example is Pythium. In gametangial copulation, the protoplast of the gametangia flows into another through a pore. So here it is the entire fusion of the protoplast of two gametes. Example is Rhizopus. Spermatization. Here, minute conidia-like gametes called spermatia come in contact with the receptive hypha. This represents spermatization. The example is Puccinia. In somatogamy, the fusion of two mycelia which belongs to opposite strains takes place, and the example is Morchella. So these are the two hypha of opposite strains and they fuse to form a new mycelium or new fungal cell. Next, we'll move on to the classification of fungi. We will see the Ainsworth's system of classification, which was given in 1971, and Ainsworth proposed a natural system of classification of fungi based on morphology and especially the reproductive structures. So, kingdom fungi is divided into two divisions, Myxomycota and Eumycota. The Myxomycota division is further divided into four classes, namely Acrasiomycetes, Hydromyxomycetes, Myxomycetes, and Plasmodiophoromycetes. Eumycota is subdivided into subdivisions, Mastigomycotina, Ascomycotina, Basidiomycotina, Zygomycotina, and Deuteromycotina. Each of this subdivision is divided into classes. Subdivision Mastigomycotina, which has motile cells present and perfect spore and oospores. So the four classes are Chytridiomycetes, Hypochytridiomycetes, oomycetes, and Plasmodiophoromycetes. Subdivision Zygomycotina has perfect spore called zygospore, and their two classes are Zygomycetes and Trichomycetes. Subdivision Ascomycotina has a perfect spore called ascospores. 6 classes belonging to this Ascomycotina are Hemiascomycetes, Loculoascomycetes, Plectomycetes, Lobulbaniomycetes, Pyrenomycetes, and Discomycetes. Subdivision Deuteromycotina has perfect stage absent, and the three classes are Plastomycetes, Chylomycetes, and Hyphomycetes. Subdivision Basidiomycotina has a perfect spore called basidiospores. Three classes are teleomycetes, hymenomycetes, and gastromycetes. This is all about the Ainsworth classification system, and these are my references. These are web links. Thank you, students.